Hello, class. Today we will be learning about the 50 states. We got Oklahoma, Texas, Mexico, New Mexico, Arizona, Colorado. But then we will be learning about glycolysis and the Krebs cycle and pyruvate conversion. And Massachusetts. Now that we're done learning about the 50 nifty United States, we're gonna head on over and learn about cellular respiration. Can anyone define cellular respiration? Anybody? Anybody? Hold up! Yeah! Do you want to learn about cellular respiration the fun way? Wardrobe! Good job. Well, head on over and we'll find out together. Yes! So, I'm ATP, and this is my friend the cell. How you doing, cell?
I'm just swelling the cell, ATP.
Well, that sounds good. I bet you're wondering to know where the ATP molecules are located. Truth is, they're located in the mitochondria, right there, powerhouse of the cell. In aerobic respiration, it's told that 38 molecules are produced at most, but the truth is, usually about 36 are produced at most. That's the fact.
And now you know.
Aerobic respiration is cell respiration in the presence of oxygen. Oxygen is a key component in aerobic respiration because it is the final electron acceptor. It allows additional electrons to pass through ETC and allows ATP to continue being produced. That's a fact, and now you know.
Glycolysis consists of 4 steps. Step 1. 2 phosphate groups attach to glucose and the new 6-carbon compound is supplied with 2 molecules of ATP. Step 2. The new 6-carbon compound splits into 2 molecules of PGAL. Step 3. The PGAL molecules are oxidized and each receive another phosphate group. Also, NAD+ is reduced to NADH. Step 4. The phosphate groups are lost from the PGAL, which produces 2 molecules of pyruvic acid, which is used in the Krebs cycle. The lost phosphate groups attach to 4 molecules of ADP to form 4 molecules of ATP. This results in a net gain of 2 molecules of ATP. When pyruvic acid comes into the mitochondrial matrix, it mixes with a molecule called coenzyme A, or CoA. When they react, a molecule of carbon is lost from pyruvic acid, making acetyl-CoA. During this conversion, NAD+ is reduced to NADH. This whole process is called pyruvic conversion. The Krebs cycle breaks down acetyl-CoA to produce CO2, hydrogen, and ATP. There are 5 steps of the Krebs cycle. Step 1. A 2-carbon molecule of acetyl-CoA mixes with a 4-carbon molecule of oxaloacetic acid to make citric acid. This reaction also regenerates CoA. Step 2. Citric acid loses a carbon and hydrogen, making a 5-carbon compound by losing an electron. Citric acid is oxidized and the hydrogen atom reduces NAD+ to NADH. Step 3. The 5-carbon compound loses another carbon to make a 4-carbon compound. The compound also loses a hydrogen. NAD+ is reduced to NADH once again. Also, a molecule of ATP is synthesized to a molecule of ADP. Step 4. The 4-carbon compound loses a hydrogen to reduce FAD to FADH2. Step 5. The 4-carbon compound releases a hydrogen to remake oxaloacetic acid, which keeps the Krebs cycle going. Also, NAD+ is reduced to NADH in this step. The electron transport chain is the second phase of aerobic respiration. Energized electrons from FADH2 and NADH travel through a series of molecules. As they travel, they lose some of their energy, which is used to pump proteins from hydrogen to the other side of the mitochondrial matrix. The concentration of protons outside the matrix is much higher than inside. This creates a concentration gradient. This helps the synthesis of ATP happen. The ATP synthase turns ADP into ATP when protons move down the gradient back into the matrix.
Anaerobic respiration is cellular respiration when oxygen isn't present. In the absence of oxygen, cells can convert pyruvic acid into other compounds. This process is called fermentation. In fermentation, ATP is not produced, but cells can produce other compounds that can be used throughout the cell. That's a fact, and now you know. One type of fermentation is lactic acid fermentation. In lactic acid fermentation, pyruvic acid and an enzyme turns pyruvic acid into another 3-carbon compound, lactic acid. During lactic acid fermentation, hydrogen atoms from NADH and H+ are transferred into pyruvic acid. This oxidizes NADH to produce NAD+, which keeps glycolysis going. That's a fact, and now you know.
In alcohol fermentation, pyruvic acid is converted into ethyl alcohol. This pathway requires 2 steps. In the first step, a CO2 molecule is removed from pyruvic acid, which leaves a 2-carbon compound. In the second step, 2 hydrogen are added to the 2-carbon compound, which forms ethyl alcohol. That's a fact, and now you know. Pass. Fun fact. Did you know a sparkling wine, such as champagne, has CO2 that is retained to create a carbonated beverage? True. 1 shot off the backboard. Oh, one more. Just one more. Woo!
Cellular respiration is fun!
Follow me along and we shall learn.
Not present.
Why don't you look like you're...
Other compounds, which I forget.